Hello everyone, I just want to talk you through what's going to happen in Lab Session 7. So this is kind of a capstone lab for us. You've learned a lot of basic lab procedures, from just aseptic techniques to wet mounts, and then you went on to gram stain and endospore stain and thinking about streak plates and colony morphology. So Lab 7 is an opportunity for you to take what you learned in the previous labs, which were mostly TED Ed with some Labster labs, and I'm going to give you some results for an unknown bacterium. So first I want you to take a look at the Lab Session 7. The background reading is going to give you all the information that you need as far as, again, conceptualizing what we would do if we were in a face-to-face lab. So like here it says unknown day 1, you would be given an unknown bacterium on a nutrient agar slant. And then the first thing you have to do is make a streak plate of that. The next day you would come in and you'd make a fresh culture and you would do a gram stain from that fresh culture, and then you would move on to inoculate various nutrient broths, including phenyl red lactose, phenyl red mannitol, anyway, glucose. So these are different carbohydrates for your unknown bacterium. And you would also inoculate a citrate agar slant, methyl red broth media, and a starch plate. So we know that you're not able to come to lab and do that with us. So what we have done is done that for your unknowns and given you some results. Okay. So if we just kind of scroll down a little bit in this lab, it would say, you know, whenever you check your tubes and so on, what you're going to look for. You're also going to do an endospore stain, and then you get to this part here. Okay, so don't gloss over this lab because this is going to give you all the background information that you need. So here it's going to tell you about those carbohydrate fermentation tubes. So you might remember I mentioned glucose, lactose, and mannitol. Very common carbohydrates that bacteria like to use. My computer's being slow. As you see here, we've given you some information in a table on how to interpret your broth tubes, and then we've included a picture. So this is going to help you know what to look for. So for example, for the results for the carbohydrate fermentation, this tube just represents the uninoculated tube, so that's your negative control. A positive control for acid shows us this yellow color change that occurred in that test tube. And then here, not only do you see the yellow color change, but you also see that there's an empty space at the top of what we call a Durham tube. So that means that your bacterium made a gas, and that gas bubble accumulates in the top of this inverted tube. So you're going to be using this information to help you figure out your bacterial unknown. Next, we have the citrate utilization test. Again, read the background information. Here's possible colors of your citrate test, and then we put the controls down here. So it says if your citrate agar slant has a pH of 6 to 7.6, then that's negative for your bacterium being able to utilize citrate for its energy source, so it's going to be green. So if I scroll down, you see this picture here. So this is the agar slant that you would have transferred some of your unknown onto. And then you see the second one, or the one in the middle, is an inoculated test tube, an inoculated agar slant that's a negative control. So this bacterium was not able to use citrate. And then the one on the far right, this one was able to use citrate. And so if you go back up to the table, then you're going to see that the pH must be greater than 7.6, and so that caused a color change in our test tube. So it mentions here that if your organism is able to use citrate as a carbon source, then it's going to produce alkaline products, which raises the pH, and it turns bromthymol blue from green to blue. So that green negative control becomes blue. All right, So then you go on down and you got methyl red, and you're going to see here again we have some controls for you. Scroll down a little bit more. Here's starch hydrolysis. So what we would have done here is you would have taken the inoculating lube and just make a single line down your agar plate. And then after it grew up overnight in an incubator, and depends on what your actual bacteria looks like on a petri dish, but here's a couple different examples, but this is just showing here's a negative control. So this is what it would look like when we pulled it out of the incubator. And then you're going to flood the plate with iodine. And it really helps to take a look at this. This is on a black surface, but you see that it basically looks the same, just now you've got iodine all over it. Now this is a starch plate, and iodine is a positive test for starch. So that's the reason why it looks kind of a rust brown. Over here, the plate was flooded with the iodine, and you're going to see if you zoom in that there's a little bit of a halo around this bacterial line, because this bacterium was able to digest the starch in the agar plate, and now when we flood it with the iodine, it appears like a halo because there's no starch there for the iodine to react with. So do please read the directions, and if you get confused on any of this, just ask me questions. You're not learning by yourself, although you might feel that way, just like I feel like I'm teaching by myself. This one's kind of fun. So basically it says catalase is an enzyme that breaks down hydrogen peroxide into water and oxygen. So if your organism can make catalase, then it's going to break down hydrogen peroxide when we put some drops of hydrogen peroxide right on top of your bacterial colony. So here's an actual dropper bottle full of hydrogen peroxide, and it's kind of hard to see, but we put some hydrogen peroxide on this plate, and you don't see any bubbling. But over here, you see it looks like maybe like your cat, or in our house, your ferret, threw up, and it looks kind of bubbly like that. So that shows us that your organism is making catalase, and it's breaking down hydrogen peroxide and making bubbles. If there are no bubbles, when you take a look at your results, then you know that it's negative for catalase. Then there's a super duper thing at the end called the dichotomous key. So this is going to seem a little whack, but basically what you have are two sheets. The first sheet's all the gram-positive bacteria that your unknown could be, and here's all the options if you have a gram-negative bacterium. What I have done is created a result sheet for you, and it'll give you a picture, and then you have to look at it and determine is it gram-positive or gram-negative, that sort of thing. And once you've done that, then you know which sheet to start with. If it's gram-positive, then you're going to ask yourself, does my bacterium have coccus-shaped cells or bacillus-shaped cells? Let's just say you say bacillus. Now, it gets a little confusing here. Now, this is kind of a yes-no thing. Does your bacterium make catalase? If it was positive for catalase, then you're going to go down this side of this branch. And if it was negative for catalase, then you go down the right side of the branch. And then we have a different question. So let's just say your bacterium was negative for catalase, so no bubbles. Now you're here, and it's going to ask you, what was the result from the carbohydrate fermentation for glucose? And this is asking about gas. Did you have gas present from carbohydrate or from glucose fermentation? Then you would go this way for the positive. And if you did not, then you would go here to the right. So let's just take the negative, just so we can go a little bit further. Now we have acid made from mannitol. So again, that would have been that color change to yellow in your mannitol fermentation tube. And if we scroll down a little bit, the pluses and minuses are down here. So let's just say that yours did not, forgot what I said, that your organism did not make acid from mannitol. So therefore, we're going to go with this last choice, and that's Lactobacillus acidophilus. So this is a bacterium that's very common in yogurt. Now, if you had started with the gram-negative bacterium, then you start on the next page, okay? And you do the same thing. So citrate, positive for citrate, negative for citrate. Here, lacid from lactose, positive and negative. Okay? So I know it's a lot to take in. I'm going to put together a little bit of a TED Ed lesson just to kind of, get you primed for thinking about doing this. And then I will email you all your unknowns because you have different ones. So it's kind of like a little puzzle, like a mystery. So if you have questions, let me know.